Thank you.
30 minutes.
Okay. Well, thank you. I would like to thank you, the organization for the invite me to talk of our work in some cars. I'll speak about a new project that I'm working on since a year and a half ago, which is using the optics techniques for agriculture. Okay, we started like a year ago. We started this new lab at the optical group. It's called Lab of Innovation on Agriculture and Ophthalmology, but ophthalmology is for historical reason. But it's basically a lab for innovation in optics for agriculture. This lab belongs to the optical group in the Institute of Physics of São Carlos. What is the idea of this lab? We think, we know a little bit of optical techniques, and Brazil is a very important player in agriculture worldwide. The next presentation, this will be very clear, but I have one slide showing how strong is Brazil in agriculture. Brazil is like the seventh large country in the world, but in agriculture we are in the third place. First place is China, then United States, and then Brazil. Okay, so Brazil is the third largest producer of food, and it's more important than that. There is a lot of land that are still to be used. Okay, so we still have a long way to go in agriculture and to get maybe to the first position. Okay, so the idea of this lab is to explore optical techniques in agriculture. There are not many companies or labs in the world doing that, using optics for some application in agriculture. And we had to choose one specific culture to start with, and we start with citrus, orange culture. Why we start in orange? Because in orange, Brazil is the largest, by far the largest producer of orange in the world. We produce basically 31% of all orange market in the world. And more than that, all this production is in the state of São Paulo, so it's nearby for us. Okay, São Carlos is right in the center of the state of São Paulo, so it's very easy to go to those orange plantation. Okay, so our lab, the first technique that we start with in our lab was in the citrus, and what problems we have in the orange plantation. There is a big threat to the orange, which is it's called the greening, or HLB. This is a very bad disease for the orange, because there is no cure for greening. You have to eradicate the trees. So the orange trees lives for like 10 or 20 years, and after you got disease, you have to eradicate, to cut the tree. Okay, so this is a problem for orange, the HLB. this disease is caused by a bacteria, and this bacteria is it's inoculated by this guy. This is a piscillid. It's very small, like 2 to 3 millimeters fly, and the big problem is that this disease, the greening, you only see after 1.5 years that the tree is already with the disease, and the tree is with the disease. You don't see, but this guy goes from one tree to the other, and it propagates the disease. Okay, so this is very bad, and if you can detect this disease before before you can see in the asymptomatic phases, it may. I'm going to show that it may make a lot of difference in treating this disease. Okay. The origin, whenever the tree is with this disease, the origin is like that, and there's no use. If you cut the origin, it's like it's not orange. It's like looks like a lemon. Okay. Anyway, so this just some economic facts for the greening in Florida. This disease started in China, and China do not produce any origin anymore because of this the greening. And in Florida, this is 205 numbers, but today almost 100% of Florida origin is with greening. Okay, so the numbers in 2005 is 4.5 billion economic damage, jobs, 8,000 jobs lost. In Brazil, 27 million trees are eradicated in 1 year, and the production loss is about 5 to 15% production. Oops, what happened? Oops. The way to see this, the greening, it's amazing, but it's some of not all the leaves of the tree become it's like a yellow, becoming a little bit yellow. It's very hard to see, and the way to see the greening, you get a tractor and 4 people, the driver plus 4 people in those platforms. So 2 here and 2 here, and this tractor is all day going. Actually, there's 4 in the farm that we go. There's about 4 or 5 of those tractors going all the way back and forth in those lines, trying to see some yellowish leaves that may be greening. Okay, so you have like a 5-person each tractor, and 5 tractors, so it's 25 person going all the way back and forth, all the day back and forth. And whenever you detect, you have already 1.5 years that the disease is being propagated. Okay, but this is the only way to control today. So this is a visual inspection. 60% accuracy, because most of the time you don't see, most of the 45 to 40% of the time you don't see. Okay, and you only see symptomatic phase, symptomatic trees. The other way to see is called PCR, and the PCR is very precise, but it's very costly, and it's unfeasible logistically. You got to get the leaf and go to a lab, do the PCR, takes 20-30 days, and you have millions and millions of trees in the farm to look for. So this is unfeasible, and so we decide we decided to start a project to detect this disease on the farm, not in the lab. Okay, the lab we did, I'm going to show you, we already see easily. So the idea now is to go to the farm. Okay. This is a slide that I just show you. The difference between starting the detection in 1 year, with the delay of 1 year in starting the detection in the asymptomatic phase of the disease, if you get up to 2020, you get like 73, you save 73 trees to be eradicated. Okay, so it's a big economic change if you can do that. Okay, so that's the project. That's the problem to work for, and so we did experiments in the lab. I'm going to show some results, and I'm motivating my students to start this company. It's called AgriOS, Agriculture Optronics System, and the mission statement is here, and the team. Those are 4 students of mine, electrical engineer. This guy specializes in hardware, electrical engineer, specialized in software, mechanical engineer, and physicist. It's interesting because I believe those type of company, high-tech company, you have to have a multidisciplinary team. There is no, I believe it's hard for one person to know all the aspects of the project. So it's a team that you have from engineers to physicists. Okay. The lab experiment, actually this one was done at Embrapa in São Carlos. The lab luminescence was made with you have like a laser here, and the laser you shine this laser to the leaves, and there is fiber optics that get back to to a spectrometer inside, and you connect this spectrum to a computer to do the analysis. Okay, basically the experiment, the diagram is like that. You have laser here. Then if you have the fluorescence, go to a spectrometer, and the spectrum you send to the analysis. Okay, so what we did. Let me show some results first. This is the fluorescence spectrum of the leaves. Okay, basically this is the fluorescence of the chlorophyll. There's two phase 1 and phase 2 chlorophyll, and there are some other things here. So it's visible and a little bit in the infrared. And what you see is that the healthy trees, you have less fluorescence in the visible, and the first peak is a little bit higher in 700 nanometers, and the second one it's also, I mean, this one is higher. Okay, whenever you get to the symptomatic, you see that this second peak goes lower, and this one goes higher. Okay, increase a little bit, but you can see here that the asymptomatic you can see it. Okay, you have here a way to see this symptomatic phase of the disease. Okay, so this one is just the laser. Okay, so if you put this spectrum in a neural network, you see the principal components here, and you see that you can discriminate very easily healthy, asymptomatic, and symptomatic. Okay, so you can diagnose the asymptomatic phase of the disease. Okay, with 92% of success. Okay, and this is showing whenever you're using those visual inspection, you start to see the disease after 1.5 years. Okay, and this is an experiment that you inoculate the tree and look for every day. You look for trying to see the disease. So visual inspection after 1.5 years, and with this luminescence, you basically see from the first day the detection. Okay, and if you look statistically, let's say I start today this detection of the trees. This is how the greening is progressing in the farms. Okay, today we have almost like 20 to 30% in Brazil of infected trees. And this will go to 100% in 2016 or 2017. Okay, and the lower graph is the eradicate trees. Since you eradicate today after 2 or 3 years, so it's about 30% of the total trees are eradicated. Okay, so whenever you get to 100%, 30%, about 30% are eradicated. Okay, millions and millions. If you start detection today, what happens is that in the first year, you eradicate much more trees than you would eventually do if you don't detect. So you see this graph, it goes up, and because you eradicating all the trees in the asymptomatic phases, and then you start to go down and you control the disease. If you do not detect in the asymptomatic phase, you go up and you get full infection of all the trees. Okay, and just to show you what results are that can be achieved. This is what happens if you the red is if you have the visual detection, and the number of orange collected. This is box of orange. Okay, 400 million. Today is about that 500 million. And then you start to decrease, and then this is like 2020. If you start detection today, asymptomatic phase, the number of trees start to go down, and then you get the control of the disease and start to go down. And the difference in the two scenarios is about 300,000 millions of boxes of orange. This is a few billions of dollars. Okay. So how is our equipment? So this tabletop fluorescence experiment, it has to be in the hand of a person. Okay. So this is the equipment. Equipment is like the size of a credit card machine, and you have here the laser was replaced by a LED. The spectrometer, that was a big tabletop spectrometer, is a small, like a small box like this size, and you have to have the processor to do the neural network calculation and show the spectrum and show the result on the field. Okay, so you go to the tree, take a leaf, measure, and show infected or not infected Okay, so this is showing just basically how is the display, the machine, the display, and this is measuring. Okay, we have agreement with the largest producer of orange in Brazil, which is basically the largest producer in the world. And so we go to the farm, get leaf, measure, take leaf, measure, and say, show infected or not infected. Okay. If it is infected, you have to glue some tape, and there is one guy that can cut it. Okay. All right. So you see, orange, nice orange like this, but this tree can be infected, and you don't know. You're gonna see after a year and a half. All right. Okay. We got some grants for starting the company from FAPESP, and measurement today, the phase is to measure. I mean, we develop in the lab some the basic principles. Embrapa, lab in USP, and then we those people from Agris develop the full equipment for measuring in the. And this also, the battery of this equipment have to last the whole day of measuring. Okay, and everything is working fine. Okay. So what is next for this company? There are other diseases in orange. There are a lot of other type of crop type, for instance, sugar canes and so on. They are integrating this system to management system, farm management system. And one thing that's very important for us today, we are working on 3 other techniques that we are working in the lab, and leaps for soil diagnosis, runway spectroscopy for maybe disease we don't know yet, and OCT for looking for plants, for fruits after they have been taken from the tree. Okay, so there are other perspectives going on, other techniques. This company, like I said, the AgriOS, we start like a year ago, and we hope that this company will get to a point of a company that I started in '86. It's called Optronics. Optronics started in '86, and same thing from the Optics group. And today, in 2010, Opto had about 400 employees. The production plant in São Carlos, this is the production plant. Okay. And we have sites. I am part of Opto. I was the founder, and I still am part of the company. So sites in São Paulo, Fortaleza, Porto Alegre, Mexico City, and Miami. And what Opto does, develop and produce high-tech equipment for ophthalmology, development and production of satellite cameras for the Brazilian space program, development and production Optronics for the Brazilian defense program, and the annual revenue in 2010 was approximately $45 million. Okay. Sorry. Okay. I made a last slide for you. Interesting one. How is the evolution of those companies, starting from one single institution, University of São Paulo and São Carlos, and then what started was here, the optical group in the 80s, and then the optical group, a lot of company in optics, just company in optics, okay? A lot of companies started from this optical group in the Instituto de Física de São Carlos of USP. So in the 80s, Opto and Industria, and then in the 90s, High-Tech, Semaco, DMC, and Alta, ARTech, Optics. And then in 2000, there's a lot more, WaveTech, and 2010, we are working on this. AgriOS is here. Okay, so I was involved in some of those, Opto, High-Tech, Optical Line, WaveTech, AgriOS, and Gene Solution. Okay, so I was involved in the process of starting those companies. All of them are spin-off from the optical group, but the process got out of control. That's all that we want. Okay, and more people are involved. We are not. I mean, I don't. Some of those companies, I don't know exactly what's going on, but they're doing well. And what we expect is that this slide in 20 years will be full of several companies in optics. Okay, so that's about it. Thank you.